Okay, welcome back to part 2 of chapter 13, discussing gene mutation. So all of those molecular and functional types of mutations we have talked about only involves one mutation, but sometimes the expression of one gene or one mutation can be affected by a second mutation. This is called suppressor mutation. Okay, this is where the second mutation actually hides or suppresses the expression of the first mutation. Now, don't confuse this mutation with reverse mutation because they are two different things. Now, you have the wild type here, for example, that makes red eyes for fruit flies. Now, forward mutation means that you are creating or turning the wild type into a mutated type. So from red eyes into white eyes, that's a forward mutation. Reverse mutation, we're looking at that same gene. Seems like A is the gene here that went from A positive wild type to A mutant. That's what led to the white eyes. So reverse mutation means we're switching the A mutant version back to the A wild type version of that gene that controls the color of the fruit fly's eyes. Where suppressor mutation, we're talking about a second gene that is affecting the expression of the mutant phenotype. The mutant phenotype was white eyes. Okay, so we have mutated both A negative, okay, which is supposed to be white eyes, but somehow a second mutation in the B gene, which gives you that B mutant gene, have turned that white eyes back into red eyes. Okay, so this is different from reverse. Reverse mutation is affecting the same gene, same location of the original mutation. You're fixing the original mutation. Suppressor mutation, you're doing something else on another location that affects the expression of your mutated gene. Okay, again, this is why in the yeast lab, when you're explaining why the red colonies turn into white colonies, you probably have mentioned that it may be due to a reverse mutation or a suppressor mutation. Now you understand what those were about. Okay, so remember not to confuse suppressor mutation with reverse mutation. Okay, and that suppressor mutation always occurs at a site distinct, okay, or different from the site of original mutation, and this is what we call them as double mutant. Okay, suppressor mutation always involves a double mutant, meaning that they have two mutations, the original mutation and the suppressor mutation that give rise of the wild type phenotype, not a reversion of the original mutation. Okay, so with suppressor mutations, there are two different categories. One is called intragenic suppressor mutation, which is when you have the original and the suppressor mutation on the same gene or codon. Now, this can be actually happen in three different ways. One way is when the double mutant happens within the same codon. So this is when the double mutant happens within the same codon. Okay, so this is what's being illustrated here. So for example, you have your original wild type sequence here that makes this particular mRNA, and then it also encodes for leucine. Okay, you have a missense mutation of your base substitution, which then changes your AT here to TA instead. Now for mRNA, you have UUU instead, and now you are encoding for phenylalanine instead of leucine. So an intragenic suppressor mutation might be where you now have a second mutation in the first base of that codon. Okay, the first base of the codon was switched from TA to CG. So now your mRNA sequence reads CUU. Even though this particular amino acid sequence is different from that one, it happens that both of these encodes for the same amino acid, leucine. Okay, so this is your original mutation. Okay, and this here is your suppressor mutation. Okay, so in a sense, this is a double mutant. Okay, double mutant. You have a first mutation that led to a different amino acid incorporation, but because you have this suppressor mutation, I like to call it as the rescue mutation because it rescues the original mutation and turn it back to the original amino acid that you're supposed to have in your protein. Okay. All right, a second way that their suppressor mutation, intragenic suppressor mutation will work is where we have an insertion or deletion, for example, from the original DNA sequence. It messes up the reading frame as we have discussed, and you end up with a frame shift mutation. Now, a second suppressor mutation can actually put that back into the correct reading frame. So, for example, you have this original sequence, okay, with the DNA sequence, and then the consequent mRNA sequence, and then the consequent amino acid sequence. That's what your wild type sequence is supposed to be. And then, let's say there was one nucleotide deletion. So, this particular T right here was deleted in this particular original mutation. Okay, original mutation. And now that T is deleted, so every new codon after that was reshifted, frame shift mutation. So from valine on, every one of these amino acids is now different from the original sequence. Okay. Now, if you have a suppressor mutation somewhere else where you insert back a nucleotide into the sequence, you took out the T, but now you insert that extra T back in. So you took out the T in the original sequence, but you also insert it right back, not too long after that. And now what happens is you inserted the T, so every codon after that will not be regrouped into your frame shift mutation, okay? So you still have phenylalanine as your first one. You have two different amino acids, but everything after that at least is the same as the original sequence, okay? So even though the suppressor mutation still created two different amino acid in the sequence, this is definitely a much less severe consequence than changing all the reading frames for the rest of the sequence. This situation here is much better than this situation here, okay? Now, in the same way, an original mutation due to an insertion may be suppressed by another deletion in the same gene. So just remember that when we're talking about intragenic suppressor mutation, other than we can have one within the same codon, we can also have in terms of when you have your original mutation is an insertion, it can be rescued by a deletion somewhere else. The other way around is also true. You deleted a sequence from your original mutation, and then it can be rescued by a second suppressor mutation that inserts the sequence back in, okay? So that you shift the reading frame back to the original wild type sequence, okay? All right, now example 3, a third way suppressor mutation can work is by affecting the overall protein structure and shape. For example, here you have your normal DNA sequence and then your normal mRNA sequence, and you have your normal protein function, shape, and structure, okay? Now, your original missense mutation somehow switches and affects the protein structure quite a bit, so now your protein structure looks completely different. Now, with suppressor missense mutation at a different site, okay, this may actually recreate the original folding of the protein by restoring those amino acid sequence, okay? So the original missense mutation led to this misfolding of the protein shape and structure, but a second suppressor mutation somewhere else, maybe 100 amino acids down the road, somehow affects the protein structure in a way that it folds it back into the wild type shape protein structure. This can happen as well. So and sometimes you're talking about a suppressor mutation on different types of RNA molecules, as we will discuss in a later on in the next example. But it is a different type of mutation somewhere else that can affect the protein structure, fold it back into the right shape. Okay, so understand the difference between these three examples. First example, you're affecting the same codon where your first original letter change was reversed by a second letter change. The second example, you're talking about where you inserted something the second time you deleted something back, okay? Or you deleted something and you insert something back. Now, the third example, we're talking about the protein function effects, protein structural effect. One mutation unfolds a protein a certain way or in a different way that makes it non-functional, but a second mutation somewhere else folds it back into the correct structure. Okay, so understand the three ways that this can be different in terms of intragenic suppressor mutation. Within the same gene, a second mutant has rescued the phenotype. Okay, now the other type of suppressor mutation is called the intergenic suppressor mutation, which is when the original and suppressor mutation are on different genes. Okay, intragenic suppressor mutation was the same gene and in three different ways. Intergenic suppressor mutation, you're talking about somewhere else in your genome, okay, that can affect the mutation's phenotype. Okay, so your original and suppressor mutations are on different genes or sometimes even on different RNA molecules, which is what this particular example in the diagram is trying to illustrate. Okay, so this is your wild type sequence where you have your DNA sequence, mRNA sequence, you have your nice protein sequence, primary structure there, you get your full functional wild type protein. Well, there was a original mutation, which is a base substitution. Then it led to an early stop codon. Okay, well, what kind of base substitution, what kind of functional type was that when you switch a stop codon? Missense, okay? Excuse me, nonsense mutation, okay? Nonsense mutation. So you have the, it makes that stop codon occur early within the sequence. So now your polypeptide protein, okay, stops in the making, of translation. So now you have this short little dinky protein that has stopped translation, and that's why you have a truncated version of the protein. So, an intergenic suppressor mutation, for example, of course, there can be many ways this intergenic suppressor mutation can happen, but this is one example. Let's say that somewhere else within the genome encodes for your tRNA sequence. Remember, tRNA needs to be transcribed as well, okay? And this particular tRNA sequence had an error in it. You have a mutation right there that instead of having an AUA as the anticodon in the tRNA sequence, now you have AUC in the tRNA sequence instead. Well, had it been AUA, it might not form with UAG, okay? But UAC, okay, would definitely make this bonding very strong, okay? So this particular tRNA now will bind to that stop codon, which is not supposed to happen. Remember that when the translation reach the stop codon, it's supposed to start the termination process of translation where release factor binds to that stop codon and everything falls apart. Ribosome falls apart, the entire translation apparatus falls apart. But now you have an actual tRNA that brings in amino acid because of this mutation right there in its anticodon sequence. And hence, instead of stopping the protein translation process, this has brought in a amino acid, tyrosine, okay, in there, and then the protein continue chunking down the road and continue the normal translation process until it reaches its actual termination sequence, which is your actual stop codon. So you end up with still the full length protein that's done after translation. But you do have that one difference in amino acid. Rather than a leucine, you have incorporated a tyrosine. But this mutated protein is definitely a lot better situation than this mutated protein. This protein is definitely not going to be functional for whatever it serves. Okay, so this is gone. But this one, at least you have the full length, and hopefully that one amino acid won't affect the overall protein structure so much that you make it non-functional. Okay? All right. So we have finished talking about the types of gene mutations there can be molecularly and functionally. But the next question to ask is, where do these mutations come from? How do we acquire mutations? Well, it turns out that mutations can arise either spontaneously or induced, okay? Either through chemicals or other environmental means, and we'll go through different types of induced mutations, but we'll first go over what where spontaneous mutations come from. So spontaneous mutations refers to those that arise from normal or random conditions, something that is out of our control. There's nothing we can do to avoid it. It just happens, not because we made it to happen. And there are a couple of ways spontaneous mutations can come from. Remember that base substitution was when a wrong nucleotide was introduced into the DNA sequence. This mispairing often comes from the concept of wobble. Okay, so this when we're talking about spontaneous mutation, base substitution, there's a mispairing that happens somewhere along the way. And remember when we mentioned about wobble, the concept of wobble in translation chapter, okay, it talks about how there's that flexibility of different nucleotides coming together, that it doesn't have to be your normal Watson and Crick complementary base pair. Because that third base is a codon that's more flexible with which the nucleotide binds with. So it also can happen with normal DNA sequences, not just translation. Wobble can also sometimes randomly happen with normal DNA sequence where a nucleotide was more flexible. Okay, and instead of our usual Watson and Crick complementary base pairing, it pairs with another non-complementary base or even modified bases, those that are protonated or methylated, who knows? Sometimes there's all kinds of different special rare bases that can happen. So for example, here you have your usual TG base pairing, which is your Watson and Crick complementary base pair, but randomly, spontaneously, sometimes your cytosine will combine with an adenine, okay, a protonated type, okay, where it starts, it protonated a particular part on the adenine, which make it able to form this hydrogen bonds with a base that is not supposed to, okay? So this usually leads to what's called an incorporated error. Okay, so when you have a base substitution that leads to a mispairing, this will lead to incorporated error. So the further consequence of an incorporated error is that it may turn into what's called a replicated error. Okay, what do we mean by that? So here, it's easier to explain with the diagram. So with this diagram, again, you have your wild type DNA sequence. During replication, the 2-strand splits, okay, into your 2-template strand right there. This one went on to do replication normally, okay, and continue on with its wild type sequence. This one, okay, wobble happened, okay, wobble happened where you have now a T with the G be due to wobble, okay? So now when you split this molecule into a second round of replication, you have your normal sequence TTCG, but in this other sequence now you have AGGC, okay? It's supposed to be AAGC, but now it has turned into that AGGC. This strand here, it's wild type sequence. So of course, it's going to continue making its wild type DNA molecule after replication. This one now is, because there's a G right there, when it goes through DNA replication, of course, a C is going to be incorporated. That's the normal complementary base pair. So now this becomes a permanent replicated error, okay? So this was an incorporated error right here due to wobble, okay? Whereas now, after a round of replication, you have incorporated in this error in there permanently, and however many times this strand is going to be replicated, it's just going to keep making that same mistake over and over and over again, and nothing is going to be done to fix it because it's a normal complementary pairing, okay? It's just that your base sequence was wrong to begin with, okay? So this is what we mean by a replicated error right here, okay? Replicated error. Excuse me for my phone's noise, so let me turn that off. Okay, I'm going to turn that off. Okay, so that's what we mean by going from incorporated error into replicated error, okay? Replicated error. Incorporated error into replicated error. So what about insertions and deletions? Where do we get those from? Do those come spontaneously as well? Unfortunately, they do. Okay, they come from what's called strand slippage, which is when one of the DNA strands forms a small loop. And this can actually happen either on the template strand or the newly synthesized strand during replication. But which strand it happens on will determine whether the sequence will end up with an insertion or a deletion. It's kind of like similar concept with the expanding nucleotide repeats that we were talking about, okay? So here you have your template strand of the DNA sequence, and this is, let's say, DNA polymerase doing its job and synthesizing this newly synthesized strand. Let's say somehow spontaneously out of random times there was a strand slippage on the newly synthesized strand. Okay, so now you have this A sticking out in the sequence. Okay, A sticking out. So then, well, our DNA polymerase is just going to continue doing its job and add on more nucleotides after that to finish the process. But now you end up with this extra A that's been bulging out, and then the next time it divides, you split this two molecule, you're going to have that extra A in the sequence. So this is how you're going to end up with an insertion, is if the strand slippage happens on the newly synthesized strand. Whereas on the other hand, let's say the strand slippage happens on your template strand instead, where your T right here was looped out. Okay, so again, your DNA polymerase is just going to do its job, adding new nucleotide to the newly synthesized strand, and then finish it off like that. But it, can't help but skip this T because it was looped out. If it's looped out, DNA polymerase is not going to read a looped out sequence. And hence, this is why when the strand slippage happens on the template strand, you're going to end up with a deletion, particularly this T deletion, okay? So that's why you can see between these two examples, whether you have strand slippage on this newly synthesized strand is how you're going to end up with insertion, okay? And then if you have a strand slippage on your template DNA strand, that's how you end up with a deletion. And of course, however many nucleotides that's looped out will then create however many nucleotides that's been deleted or inserted. If the looped out sequence was 3 nucleotides, then you're going to delete 3 nucleotides. If the looped out sequence was 6 nucleotides, then you're going to delete out 6 nucleotides, okay? All right, so this is what I was trying to explain here in these two cases, okay? This will be example 1, and then this will be example 2, okay? Now, another way insertions and deletions can arise spontaneously is by what's called unequal crossing over. And we have touched on this topic when we were discussing Robertsonian translocation back from chapter 6. Remember how we said, a big chromosome and a small chromosome translocation happens, and then they would end up with a really, really big chromosome, and then a really, really, really tiny chromosome, and that really, really tiny chromosome doesn't carry that weight, and it will just be left out when it undergoes anaphase cell division, okay, and then the small chromosome will just be deleted and degraded, whereas you have the other one will be a really huge chromosome. So in a sense, this is what it's describing, okay. I talked about how important it is when two homologous sequences are undergoing crossover or recombination. It is very critical that the two sequences are aligned perfectly. That's why in meiosis, we talked about how homologous chromosomes are aligned and make sure that they stay that way through the formation of the synaptonymal complex, because that's what makes sure the chromosome doesn't shift, okay, and misalign the homologous sequence. But if the two homologous sequences are misaligned before they do a crossover or recombination, then you will end up with unequal swapping of DNA sequences, just like in this diagram. See how this section is supposed to be perfectly aligned with this section because they are homologous sequences, but because they have shifted in their alignment, now you end up with unequal crossing over, okay? With perfect alignment, you're supposed to have no net change, okay? No net change in number of nucleotides in either molecule because the swapping is supposed to be equal. But when you have a misaligned homologous sequence, then you're going to end up with not this sort of unequal crossing over, okay? One will cross over a short segment and then another will have a long segment. Okay, so in the end, with unequal crossing over, what will result is you will have an insertion in one DNA molecule and a deletion in the other. In this case, this is where we'll have an insertion, okay? And this is where we have a deletion, okay? So that could also explain why we have insertion and deletion spontaneously that can happen. Okay, so the other way that mutations can arise from, other than spontaneous, is induced mutation. So induced mutation means DNA lesions are introduced either through chemicals or radiation in the environment. So these are stuff we can try our best to avoid and try our best to control. So those chemicals that increases the likelihood of mutations are usually called mutagens because they cause mutations. Okay, so that's why we call them mutagens. Okay. Now there are a couple different types of mutagens that exist. So one type of mutagen are called base analogs. And what these are is that they are chemicals with structures very similar to any of the 4 standard DNA nucleotides. So for example, you have thymine as your normal base here, but sometimes there's a base analog called the 5-bromouracil. Even though they're completely different chemicals, as you can see, there's a bromine there, okay? And this is a T versus your 5-bromouracil, okay? But structurally, if you look at it chemically speaking, it is almost similar between these two bases. And what happens is that DNA polymerase actually can't distinguish between normal bases and base analogs. So during DNA replication, it sees a 5-bromouracil, it's going to think it's a thymine. So then it's going to incorporate the 5-bromouracil as a thymine, which then pairs with adenine. Okay? Or sometimes a mispairing can also happen where it pairs with a guanine. So that's, of course, if it binds with an adenine, then we're safe because that's what thymine is supposed to pair with. But because 5-bromouracil also occasionally pairs with guanine, this is how we can end up with a missense mutation or base substitution, okay? So what we call this type a transition mutation, because instead of a guanine, we have now bind to a, instead of an adenine, we have now bind to a guanine, okay? They're from purine to a purine, okay? So base analogs can also, just like your other spontaneous error, okay, is it can introduce the incorporated error just like your normal base substitution, and it will go on to produce a replicated error. So for example, here you have your normal DNA sequence, strand separates during replication. Here's your incorporated error right there, your 5-bromouracil right there. Okay, and luckily this time it was bind to an A, so okay, it's all right, okay, but this is considered an incorporated error. You put in the wrong base analog in there. So now when the two strands separate, this goes on, and of course normal DNA sequence, the A is not the matter here. It's that 5-bromouracil that's the problem, okay? And let's say it occasionally pairs with guanine, and that's what happened here. So here's that guanine that was mispaired with 5-bromouracil. Had this been a T, then an A would be incorporated. But now because this is changed to 5-bromouracil, that's why you have that occasional G pairing. So when these two strands separate, now you have a G sequence within the DNA sequence. So now it becomes a replicator error, because every time this particular DNA undergoes replication, it will forever have that G, okay? And then what's going to happen during replication is it will, pair with a C normally, okay? It's just continuously going to do that and becomes a permanent replicated error because of this mispairing due to the existence of the 5-bromouracil. And this 5-bromouracil, if we don't get rid of it, it's going to continue to cause damage. If it, of course, it still binds to adenine. So if it binds and pairs with an adenine, you're safe. But the only problem is when you start incorporating the wrong nucleotide due to existence of this base analog, and you're going to end up with more and more replicated error if this 5-bromouracil wasn't getting rid of, okay? So that's how base analog can go from incorporated error and keep introducing incorporated error and then turn those into replicated error. Other types of mutagens include alkylating agents, those that conduct deamination, and a specific chemical called hydroxylamine. Okay, so this is a misspell, so it's actually alkylating. Okay, so alkylating. Okay, alkylating agent. Okay, so what happens with these is that alkylating agent, as its name suggests, is conduct alkylation by either adding a methyl or ethyl group. That's what alkyl groups means in chemistry speaking. Okay, this is an ethyl group that's added to this particular oxygen on the guanine, so that now it becomes ethyl guanine. Well, G is supposed to pair with C in the wild type sequence, but because now you have a modified base, it likes to form a pair with thymine instead. So you went from a CG pairing into a TA type of pairing, okay? Now, deamination is when you remove the amino group and create a different nucleotide again. Okay, so in this case, you're removing this NH2 molecule from cytosine and turn cytosine into a uracil. Okay, so cytosine is supposed to be normally pairing with G, but because you have changed it to uracil, now it binds with A. So it went from, again, a CG to a TA type of complementary base pairing. Again, though all of these are actually transition mutation, because we did say transition mutations are usually more common because it's easier to convert a purine to a purine or a pyrimidine to pair with a pyrimidine. Okay. Hydroxylamine is a particular mutagen that adds hydroxyl group to just cytosine. So here's cytosine, and here's that extra hydroxyl group that's been added to the amino group. So now it becomes hydroxyl aminocytosine, which originally is supposed to pair with G, but now because it has changed its chemical structure, now it pairs with adenine instead. Again, instead of CG, now you have a TA type of binding. So all of these three are a transitional type of mutation, transition mutation, because it switched from a CG type of pairing to a TA type of pairing. Now, the other way we can introduce mutation is via, not radiation. We're not there yet. Okay, is intercalating agent. Okay, that's the last type of mutagen we will discuss. Intercalating agent. These are chemicals that are able to sandwich themselves between the adjacent bases in the DNA. Okay, so in this particular picture here, you have these blue area, which represent the actual, let's say A, T, C, and G, okay, the nucleotide sequence. These yellow stuff, okay, is that intercalating agent that inserts themselves between the nucleotide bases, and that actually distorts the normal double helix structure of DNA. So that's why you have this weird looking lumpy type of DNA helix you're seeing in this picture here, because of the insertion of these intercalating agent that's messing up the overall structure or integrity of the DNA molecule. And usually when you have these insertions that messes up the structure, it actually makes gaps, okay? And those gaps can actually cause single nucleotide insertions and deletion when the molecule moves on to do replication, okay? So normally, usually intercalating agent will produce frame shift mutation because they will cause gaps within the DNA helical structure, okay, and result in insertion deletion. And remember, insertion deletion causes frame shift mutation. So out of all these mutagens we've been talking about, base analogs or any of these three types, intercalating agent is the most severe type of induced mutations you can have. Okay, you will have severe mutagenic effect if you're talking about intercalating agent, and that's why in your lab exercise last week with gel electrophoresis, there was a question that asked you about using ethidium bromide versus methylene blue. Why do we use methylene blue? It's because ethidium bromide is a known intercalating agent that can lead to development of cancer and, of course, lots of DNA lesion. Okay, so this is the reason why we want to switch it out to something that's less toxic like methylene blue, so we don't end up jeopardizing our DNA sequence, and try to avoid using ethidium bromide nowadays anyway. All right. So the last way we can introduce, at least the last way we'll talk about, is radiation, which are high energy waves capable of penetrating tissues and damaging DNA. Okay, there are two types of radiation that can cause DNA damage. One is a more severe form called ionizing radiation. These type of radiation are able to dislodge electrons from the atoms they encounter, change those stable molecules into free radicals. That's what ROS here means, are those free radicals and reactive ions. Okay, and they will alter nucleotide sequence because free radicals and reactive ions, ultimately called reactive oxygen species, they actually are toxic to the cell, and they will break phosphodiester bonds within the DNA sequence. And when you cause breakage, that's a type of DNA lesion or DNA damage. So that's bad for the cell. So that's why ionizing radiation can lead to DNA damage. And some of those examples of ionizing radiation is X-rays, gamma rays, and cosmic rays. So at least with X-rays, we're not, having an X-ray every single day of our life, every minute. So it's only for necessary therapeutic cases. But, when we're talking about astronauts that goes in space, where they live at the space stations for a long time, there's a lot of gamma rays and cosmic rays in the galaxy, in the space. So that's why NASA actually, invest millions of dollars into different research type to, what is a better way to protect their astronauts from coming back to Earth, because usually after spending some time in the space station, their risk of developing cancer is a lot higher compared to normal individual who's been staying on Earth, due to them being exposed to those gamma rays and cosmic rays in space. All right, the second type of radiation is called ultraviolet radiation, which is we're more familiar with, and something that we can become exposed to constantly. Okay, we're out in the sun too much. So this has less energy than ionizing radiation. You won't dislodge electrons and become reactive oxygen species all of a sudden, but it's still mutagenic. Okay, it often leads to formation of what's called your pyrimidine dimers, which you were talking about in your yeast lab again. Pyrimidine dimers are when you have formation of covalent bonds between neighboring pyrimidine bases like thymine, okay, and now you have created some sort of a bulgy sequence, okay. If you're thinking about three-dimensional space, these dimer, because you have created some sort of a bond right there, it creates that bend, and we call these bulky lesion that distorts your DNA configuration and structure, which then, in a sense, will block replication because you have some weird thing going on in your DNA structure. Now, maybe it blocks DNA polymerase from binding, blocks a lot of things, all those apparatus and initiation factor we're talking about from binding, and hence no replication occurs and the cell dies, okay? So this is why, they say, you're having UV radiation on, equipments that you've used. You want to kill bacteria, sterilize it, you put it under the UV light, let it become sterilized, and then use it. So this is how bacteria are killed by the UV light, is because we create all these different pyrimidine dimers inside the cell, which then blocks their replication and hence stopping the bacteria from growing on that particular material. Okay. All right, so now that we understand the types of gene mutations there can be and what causes them, is there a way to measure how mutagenic a chemical is? For example, when pharmaceutical companies came up with a new drug for a treatment of diseases, they have to make sure that that particular drug is not a mutagen, or even worse, a carcinogen, which means carcinogen means it is a type of chemical that can lead to cancer. Hence, in 1974, a biochemist named Bruce Ames, again, another very famous scientist that is still alive today, according to Wikipedia. He's 91 years old, but still, he's still alive. He developed a cheap, simple screen capable of analyzing large scales of chemicals, and it can give you an estimation of whether that chemical is mutagenic, which usually means they are also carcinogenic. Usually, if they're mutagenic, 90% of the time they are carcinogenic as well. Okay, this test is called the Ames test. So here's how the Ames test works. What you will do is you will first plate a mutant type of bacteria that has a mutation with histidine, where these type of bacteria cannot make histidine, and you plate these cells on minimal media that lacks histidine. So technically speaking, these histidine mutation bacteria won't be able to grow on that medium. Only histidine positive cells or wild type cells can grow on the minimal media. Okay, so now they're going to mix whatever compound you're trying to test for, that substance, new drug that came out, but you want to make sure that it's not mutagenic or carcinogenic. Then you add that compound, mix it with a little bit of a mammalian liver extract that contains metabolic enzymes, okay, which can either be added to the agar plate itself directly or put on a plate using a paper disc where if this was the agar plate, okay, with the agar on it, you can put a little paper disc here to allow that compound or new substance to be absorbed into the agar. So you would look for colonies that represent reversion mutations from your histidine mutant form into your histidine wild type form, okay? So if you see a lot of colonies forming within your compound treated bacteria, that means a lot of mutation has happened and a lot of reversion happened to the bacterial cells that it has went from histidine minus to histidine plus. Okay, so this tells us that from the Ames test, if you have lots of colony growing on your plate, that means it is that compound X you're testing for. So compound X is very much mutagenic. Okay, mutagenic. So formation of large amount of colonies means that that particular compound is mutagenic. Now, not a lot of substances will end up showing as mutagenic. Usually out of 100 substances tested, only very few would show lots of colonies growing. But there were in history where some promising pharmaceuticals where they had that promising drug did fail the Ames test, because if this medication is supposed to cure people from particular diseases, it would be bad if the secondary symptoms or secondary effects cause cancer in the patient. So it is critical that new drugs always use this Ames test to make sure that their particular chemical won't be mutagenic. All right, so now the last part we'll talk about is mutation rate. It seems scary, not the last part, because we still have DNA repair. So it seems scary to think about all those mutagens and how sometimes mutations can even arise out of nowhere spontaneously. We can try to avoid mutagens and carcinogens, but we can't control spontaneous mutations. So are we all doomed to get cancer? Well, thankfully not. A lot of these spontaneous mutations actually occur at an extremely low rate. This is what we call as mutation rate, which is the frequency that a wild type allele at a locus changes into a mutant allele. Okay, it is usually expressed as a number of mutation per biological unit, mutations per cell division, however many mutations per gamete, or however many mutations per round of replication. So spontaneous mutation rates will vary from species to species and sometimes even from gene to gene within the same species, but they are generally very low, okay, that, regardless what organism was studied, we find that the mutation rate spontaneously is very low, okay. Typical mutation rates for bacterial gene is somewhere in the realms of 1 to 100 mutations for 10 billion cells, whereas the mutation rate for eukaryotic genes is also not very high, 1 to 10 mutations per million gametes. And this table illustrates how, whether you're looking at a bacteria or a corn or a fruit fly, whatever it is across species, their mutation rate is pretty low overall. Okay, now who do we get to thank for this low mutation rate in natural world is two reasons. One is that we're thankful that our DNA polymerase has a high fidelity of replication, okay, which means it likes to keep the same nucleotide. It tends to want to incorporate the correct nucleotide. And remember, we talked about how DNA polymerase 3 actually has a proofreading function. So that proofreading function is going to stop the mutation rate, okay, from being high. Second thing is that there exists DNA repair mechanisms. So these are talking about how efficient our DNA repair mechanism is. So because of these two things, this is why mutation rates spontaneously tend to be very low. And hence, the last topic we'll cover for this chapter are different types of DNA repair mechanisms we can have. So there are actually a number of complex pathways for repairing DNA, but in general, they have two general qualities in them. Most of them will require two nucleotide strands of DNA because usually what happens with repair mechanism is you will remove the mutated strand and delete it out, but you need some template strand to make sure you're incorporating the correct sequence. So that's why one is usually deleted, and you need the other to serve as a template strand to tell you what is the correct nucleotide sequence. Okay? The second thing is most of these repair mechanisms are redundant, which means many different types of DNA damage can be corrected by more than one repair mechanism. Okay, this is sort of like our nature's way of protecting us. If that particular mutation escaped one repair mechanism, we always have a backup somewhere else that's going to recheck the process and try to repair it back. Okay, so if it escapes one, at least we have those other three that can do the same thing. Okay, so that's why that's another quality of our DNA repair mechanism that helps us keep the mutation rates very low. Now, there are many types of DNA repair, and they are all very complex. There's actually a whole research field that is based on DNA repair mechanisms and pathways, because in a sense, this is how we are able to study cancer and try to come up with new ways to fight it. What is it with cancer that we can escape, that made them able to escape DNA repair mechanism? Can we do anything with our current DNA repair pathways so they can kill cancer cells? All of these questions that can lead to potential anti-cancer therapy relies on fundamental understanding and knowledge of how DNA repair works. So that's why it's a complicated concept to talk about, but we will just discuss a few here to keep things simple and just to introduce you to the topic itself. We already talked about two of them in your lab activity questions with the yeast mutagenesis lab. One was called photoreactivation, if you remember. Okay, this is where your photoreactive enzyme called your photolase can use energy just from visible light to split that bond that was formed by your pyrimidine dimer, okay? And then once you split that covalent bond, then you have your normal pyrimidines pairs there, and it won't create that bulge that was existing in the original pyrimidine dimers. Now you have your normal shape of the double helix because those covalent bonds were gone, okay? Or destroyed by photolase. Again, now this photoreactivation process only exists in bacterial cells and yeast cells. That's why in your lab exercise, you had to mention a different type of repair called the nucleotide excision repair, which exists in humans and other cells. Okay, the lab the lab makes it sound like it's just in humans, but it actually exists in humans and other type of cancer as well. So nucleotide excision repair, or known as NER, is like a gene editing program, if you think about it, that we have to intrinsically fix misspelled letters. So it tends to repair larger regions of damage that distorts your DNA helix, not just pyrimidine dimers, but also if you have repeated sequence that's wrong, okay, or multiple nucleotide within your codons that's wrong, okay? Within your DNA sequence that's wrong. So what the process of nucleotide excision repair is that, first of all, you need to separate the two strands of DNA that's going to be repaired. One section of DNA containing that particular distortion will be removed, deleted out, okay, by endonucleases. Now you have this gap you need to fill in. So here comes your DNA polymerase, which will then put in the correct nucleotide again to your according to your template strand here. Okay, and then DNA ligase will come in and ligate those broken pieces of sugar phosphate backbone to make sure you form those nice phosphodiester bonds again. Okay, so again, this is what's found in both bacteria and humans and a lot of other organisms as well. It is actually one of the most important type of repair mechanism, but there are diseases out there that has something going wrong with this particular type of repair. For example, patients with xeroderma pigmentosum is a disease where this NER repair is actually defective in these patients. Okay, luckily it's an autosomal recessive disease. What happens with these patients is they usually see that they have freckle-like spots on their skin, because they actually predisposes them to any UV light will give them sort of that pyrimidine dimer, and they're more likely to leave those pyrimidine dimer errors in there because they don't have a functional NER to fix those type of dimers. So that's why they have a lot of predisposition to skin cancer when you have xeroderma pigmentosum. Okay. A specific type of NER we will discuss is called mismatch repair. Okay, so this is a specific type of NER repair mechanism that fixes errors that, talking about a mismatch in sequence in terms of nucleotides. Okay, so this is talking about how somehow this particular mismatch repair right here, GT, T is supposed to be binding with an A, but it binds with a G instead. That's why it's called a mismatch base. And hence, DNA polymerase 3 with that extra proofreading function didn't correct it, missed it. Okay, it escaped that particular normal mechanism of repair. So here comes a second repair, mismatch repair, that's going to try to fix this error. Okay, so how it does that is you have this particular distortion that's going to be recognized by particularly your mismatch repair enzymes. This mismatch repair enzymes then will form a complex that will cut out your distorted region, okay, of the newly synthesized strand. Now, how your mismatch repair complex is able to distinguish between template strand from your newly synthesized strand is looking for methylation. The old template strand is usually methylated, whereas your new synthesized strand is non-methylated. So that's what your mismatch repair enzyme or complex will try to look for. Okay, which strand has the methylation? And that's how it knows that's the strand you're not supposed to delete. Okay, they will not delete the methylated strand, and they will see it as that's the correct template strand. So they remove that entire wrong sequence out, and then now it's going to recruit DNA polymerase, fills in the gap, and goes in there and put in the correct sequence according to your methylated template strand. So now you have fixed the one error that was supposed to be GT, now it's fixed back to TA, and then DNA ligase comes in here, seals the deal, and corrects your sugar phosphate backbone. Okay, I'm sorry that I did not divide the lecture evenly between the two. I thought this last part is actually pretty quick, but it ends up there's a lot of explaining to do on the latter half, so I apologize for the longer part in part 2. So at least you have a short part 1, which was just 30 minutes. Okay, so let's go over practice question and we will call it a day, okay? So for practice question, which of the following is not a characteristic of mutations? Mutations do not provide genetic stability. Everything else is true with regards to what we talked about in mutation. Which of the following types of gene mutation in a protein coding gene usually have the least severe or deleterious effect on the phenotype? Least effect, missense mutation. Okay, missense mutation is when you are switching one amino acid with another, but this is not as bad as nonsense mutation. Remember, nonsense mutations are those that you're putting in a stop codon, truncating the protein. Base deletion, if it's a deletion, you're going to cause some frame shift mutation, which is very severe form too. Hence, all of these, for the least severe is when you just switch one amino acid, and hopefully that one amino acid is not going to cause too much of a severe effect on your final protein structure. And of course, expansion of trinucleotide repeats is pretty bad because you're having whole expanding region of repeats. We already discussed that, okay? A mutation in gene X overrides the effect of a previous mutation in gene Y and restores the wild type phenotype. The mutation in gene X is called a intergenic suppressor mutation because you have two genes involved and one restores the wild type. Okay, if this was gene X instead, then it will be intragenic, but because this is two different genes, that's why it's intergenic suppressor mutation. A blank error occurs when a mismatched base has been incorporated into a newly synthesized nucleotide chain. When this DNA is copied, this error leads to what kind of error? Okay, incorporated error occurs when you have a mismatched base, and once after replication, okay, this error will become replicated error. Okay. All of the following are causes of transition mutation except oxidation of G and its subsequent mispairing with an A, okay? All the other ones, we talked about deamination, wobble, base analog, alkylation. Those are the ones we're talking about that are transition mutation. We didn't talk about oxidation of G, so that's why that's not the one that is transition mutation. So how are old and new strand distinguished from each other during mismatch repair? We talked about the old strand is methylated, so that is why your mismatch enzyme complex can actually recognize which is the template strand and know which is the wrong or error strand to delete out and keep the template strand. It keeps the one that's methylated. In humans, somatic cell mutations are passed on to the progeny of the individual with somatic cell mutation. True or false? False. Okay, remember it is germline mutations that gets passed on, not somatic cell mutation. An organism with a suppressor mutation is considered as a double mutant because suppressor mutation means you have one original mutation and then you have one original mutation and a second mutation somewhere else or on the same gene that will revert it or rescue it. Let's not use revert so you don't confuse it with reversion, but it rescues it from the original mutation. That's why it's a double mutant. You wish to test a new pesticide to determine its potential as a carcinogen using the Ames test. You detect significantly more bacterial colonies on the treatment plate than the control plate. This indicates that the chemical tested is mutagenic and probably also carcinogenic. Remember I said how usually, oftentimes when something is mutagenic, it is also carcinogenic. It tends to it is more likely to cause cancer if it can cause mutation, okay? And that's all I have for you. This concludes chapter 13 on gene mutation and a short glimpse into DNA repair. So now remember that we will discuss the excitement of different types of modern genetic analysis technologies we have in chapter 14 and 15. So be looking forward to that.